All right, hey guys, what's going on? So today we are going to talk about a new playlist that's coming up. So I started a new playlist yesterday on GR, and that's going to be something that's going to sort of continually go on throughout the year. And now I'm starting another playlist sort of concurrently with that, also alongside the quantum field theory playlist. So if this, again, is the kind of stuff you're interested in, then you can hit that subscribe button, you can like the video, and you can go onto my Patreon page too, where more things will start coming out pretty soon here. So, and I know I keep on promising that it's going to actually start now. So without further ado, let's get into the content of this video. So this playlist is going to be a little bit more focused on the kind of stuff that I do in the lab. I am interested in ionotropic glutamate receptors. These glutamate receptors, they exist throughout the entire body. They have, they're very, very interesting, and we'll go into the reasons why they're interesting, but I'm primarily doing this because I am a working PhD student, and I think this will be helpful not just for me, but for you guys as well, to understand some more detail about some medical system. For those of you who might be interested in medicine, specifically those of you who are interested in neurology or neuroscience, this is going to be a playlist for you, because glutamate receptors are very integral to, or very important to, how we learn things in the world, how we observe things in the world, and also, very, very importantly, are an integral feature of the hippocampus, which is an organ, part of the limbic system in the brain, and this organ is responsible for memory formation, learning, emotions. And so when we get down to sort of the nitty-gritty of the hippocampus, we are forced to look at these glutamate receptors and how they contribute to the overall function of the hippocampus, right? So I'm just going to draw, and this is what is, and I'm going to be primarily going over this paper right here. So this paper right here, Structure, Function, and Pharmacology of Glutamate Receptor Ion Channels. If you don't know, we're going to break this down, this title down essentially. These are, again, these are the authors of this paper. This paper is pretty long. If I remember correctly, it's about 190 pages. It's fairly recent too, right? So 2021, right? So that was just 2 years ago now. Now that is 2023. And so this is going to give us fairly recent information. This is going to be a very good canonical overview of glutamate receptors. And I'm going to be bouncing between this paper and some other papers that are a little bit more relevant to the kind of work I do. However, if we look at glutamate receptors, right, we look at glutamate receptors, there's different types. Again, we'll get into the different types later on. This is sort of just an overview of what I want to cover in this playlist, and me talking to you about what my goals for this playlist are. And without making this too long, I want to also kind of just introduce us a little bit to the paper, and really kind of just to the title. So we're going to be looking at structure, right? So the structure of the channels, right? So what do they look like, right? The channels themselves, they're membrane channels, and they have these membrane domains on them, right? So this is just a very quick sketch. Have some membrane domains, and these membrane domains, they're alpha helices, right? Alpha helices. And then they are attached to some amino terminal domain. I'm not really drawing this very well. We have some amino terminal domain, right? So ATD is usually how this is. So the amino terminal domain, and then we have the LBDs, right, ligand binding domains, right? So these are the LBDs. This here would be sort of like the transmembrane domains, and then we have the C-terminal domains. We're going to go a little bit deeper into the structure of these, right? So this is sort of a very, very rough sketch of what this looks like, and how, and what we're going to go over the tools also on what I use, what people use in the field to study these and understand their structures, right? One of the mantras in the field, in biophysics in general, and in biology in general, is that if you have some sort of structure, some protein, there's going to be some function, right? So you can infer, say, the function of a table by looking at it. And so say you look at a table, you look at some computer or something like that, or a cup. We'll take a cup, for example. I have a cup right in front of me filled with pens. If I just had the cup in front of me and I looked at it, and I could sort of infer, well, perhaps this is something that you put something in, right? There's a function to that structure that I'm looking at. Same thing goes with these glutamate receptors. Same thing goes with almost every protein in the body, right? It has some sort of specific structure to it, and that structure lends itself well to some function, right? This is a mantra, if you will, in structural biology and in biophysics. So that's sort of the structural aspect, and then the function, right? So the function is integrally tied to the structure, right? So there's a very deep link between structure and function, just as I've talked about. And when we're talking about function, right, we're talking about measuring sort of what they do, what these receptors do. And when we measure these receptors, here, what we essentially do is we take some sort of, say, ligand, like glutamate, right? So say this is glutamate. And this glutamate is going to bind to the ligand binding domain, right? So it's going to bind to the ligand binding domain. And then when it binds to the ligand binding domain, there's going to be some sort of conformational change that will happen, right? So something binds, there's some conformational change in the protein, and that conformational change will propagate through the transmembrane domain and into the carbon or the C-terminal domain. That movement is going to propagate itself in some unique way to the C-terminal domain, and that is going to undergo, so there's going to be some conformational change here as well in the C-terminal domain. And upon that conformational change in the C-terminal domain, then you get some sort of movement going on that will initiate something inside the cell, some sort of sequence of events inside the cell. Specifically for glutamate receptors, this propagation, right, this propagation is going to result in either an opening of the channel, right, so opening, or perhaps a tighter closing, right, so, or closing, right, and we can also think about the speed of the opening, right, so say if instead this was not glutamate, but say some different sort of, glutamate is an agonist of this receptor. Say you had some sort of antagonist. We'd want to study what that does, right? So in general, we have agonist and antagonist. So an antagonist is something that will not allow for the flow of ions through, right? So, and then you have an agonist, which will. Then you have these things like partial agonists, right? So partial agonists. We'll go over a lot of this a little bit repetitive. This will be sort of a repetitive sequence of lectures, or however you want to call them, talking about the effects of all of these. And this paper again is going to nicely flesh out some of these different types of agonists, antagonists, what they look like, and so forth. A partial agonist, just to be brief, will bind to the ligand binding domain, right? Some specific movement in the ligand binding domain will again propagate itself to the transmembrane domain and to the C-terminal domain, and that is going to do something. It's going to cause the receptor to undergo some change in its conformation or in its structure, and that doesn't correspond to a full, sort of like a full current through the transmembrane domain, but it doesn't also totally close the TMD. And so what this means is that there's some sort of partial current that passes through, right? And the way you sort of measure the effects of these is through something that we call patch clamping or voltage clamping. Cells that have these on them. I'm not going to go too deep into this right now because this is really just a review or an introduction, if you will, to this type of material. But you'll have some curve, say, that looks like this, right? Where at this stage here, you apply the ligand, and then this is closing. So apply the ligand, you get some current, right? So the current is going to drive the reading of the voltage away, dramatically away from baseline, right? So that's going to be this steep slope right here. And then the channel will start closing, and as it starts closing, it takes a while for it to close, right? But it closes eventually, and you get back to baseline where you have equilibrium voltage again. We'll again go into the math a little bit deeper here, but this is what a typical curve looks like. For a partial agonist, you might have something that looks like, say, this, and maybe something like that, and then maybe something like that, right? So you can have different curves. Say something like this also. Or you can have something, say, like this, where you have a really long closing period, right? So this would be really, really long closing period. This would be really short. And this would be something in between, right? So long and then short. This might be something, this is going to be something we're going to look into as well, a little bit deeper. But this is just an overview. So that's the function. So we've covered a little bit about structure, the general gist of what these look like. We've talked about the function. And what we're going to do is we are going to, these are going to be the two main things where we're going to want to focus on, the structure and the function. And then there's the big pharmacology, right? So pharmacology is really sort of the application of structure and function within a physiological context, right? So whenever you think of pharmacology, you think of big pharmaceutical companies, right? So like Eli Lilly or Pfizer or Johnson & Johnson or something like that. The pharmacology of a receptor, or of any protein for that matter, is understanding the ligands, right? The ligands here, right? So we have a list. We can have like a list of agonists, like agonist A, agonist B, C, D, and this list could go on. And then we can have a list of antagonists also, right? Do those in yellow, right? So we have A, B, C, and D. And then we can have a list of partial agonists and so forth, partial antagonists. And we'll see that those lists, right? These lists right here of all the chemicals that sort of contribute to being an agonist or an antagonist or partial agonist to these receptors constitute the pharmacology of the receptor, right? So we can in a sense create classes of molecules, maybe even small proteins that act as the antagonists or antagonists or agonists for these receptors, and this is the pharmacology. So right, so again, I don't want to belabor this too long here because I like videos, and I think most people like videos that tend to be a little bit shorter, on the shorter end of things. But that's sort of the gist of what we want to go through, right? So we're going to go through this paper here in a very detailed way. And so again, it's 190 pages. It will take us some time, but I think it's going to really help me in understanding my field, and I think it's really going to help you in being able to understand whatever medical field you guys are interested in as well. If you're someone who is going into medicine, if you're someone who is already in medicine, especially within the neuro field, then this is going to be, I think, really interesting and helpful for you guys as well. We're going to also be talking about, just to sort of add a little bit of nuance to this, we can think of these receptors also within the context of developmental stages, right? So you could say humans inevitably start off in the womb, right? So you have an evolution from, say, blastocyst to fetus to young adult or child or baby, you could say, and so forth, right? From baby to child, from child to immature adult, and then to an adult, or something, however you want to say that. But this evolution of age, as age progresses, right, with age comes a change in the physiology. So, and when there's a change in the physiology, then there's a change in there could be a potential change in how these receptors function as well, because if your physiology is changing, if your physiology is changing, then the receptors might act and behave differently as you age. And so this is something that's going to be of interest to us also, because what we'll see later on is that these glutamate receptors, there's a subclass of them that are particularly active, a little hyperactive, if you will, in early stages of development, which is going to be very interesting for us to look at. And so, again, there's a ton of nuance here. We're going to go over this nuance. We're going to go deep into this paper, and we're going to take it slow. We're going to take it easy. And we're going to learn a lot about not just the receptors, but we're also going to try to learn about the methods that are used as well, right? So when we're looking at structure, for example, we're looking at cryo-EM. When we're looking at, here, I'll do this. So cryo-EM, maybe we'll look at X-ray crystallography. Also, for function, we'll look at various electrophysiological methods like patch clamping, and so forth. And then pharmacology, this is going to be, the pharmacology is sort of going to be an application of the structure and function within the medical context, really. That's what that is. So I don't want to keep you guys for too long. Here, I'm running on about 20 minutes, which is pretty good, I think. I'm anticipating each lesson, if you will, or each discussion here to probably be within the 15 to 20 minute range. I don't want to really exceed 25 to 30 minutes. And if this is the kind of stuff you're interested in, again, please hit that subscribe button, hit that like button, and a lot of stuff will be going on my Patreon page here on this kind of topic as well. So without further ado, I will see you guys in the next one. Bye, and have a happy new year.